Right? This figure should look really familiar. Right? We already looked at it when we were talking about neurons. So we're separating the nervous system into the central nervous system and the peripheral nervous system. And we're going to talk about the central nervous system first. So this is your brain and spinal cord. Your central nervous system is receiving information from your afferent branch of the peripheral nervous system. And these are your sensory systems. So somatic is sensations coming from your skin's surface. Special senses are vision, hearing, taste, smell. Okay, and visceral senses is information coming from your visceral organs. You don't become consciously aware of that, though. Okay. How do you know what your heart rate is? What do you have to do? You have to use your somatic sensation, right? Okay. You have to feel your pulse. Okay, output, the central nervous system gets all this afferent input and makes some sort of decision about what's going to happen in response, and then sends the signals via the efferent nervous system. Okay, and you consciously control the somatic branch of your efferent nervous system, so you consciously control your skeletal muscles. Okay, and then the autonomic branch of the efferent peripheral nervous system is not under your conscious control. It's automatic. Okay, and that's your sympathetic and parasympathetic nervous system, which control cardiac muscle, smooth muscle, and glands, and the GI tract. All right, so we are going to talk about the central nervous system in specific. So we're going to be talking about interneurons. Interneurons are housed completely within the central nervous system. Okay, so their presynaptic cell is always a neuron, and their postsynaptic cell is always a neuron. Right? This is a really simple afferent, one interneuron, efferent. Okay, usually you have way more interneurons in between afferent input and efferent output. Okay, the afferent neurons, again, the cell body is separate from the dendrites, right? But the afferent neurons, their axon terminals are within the central nervous system. Okay, and then efferent neurons, their cell bodies are within the central nervous system, and then their axons and axon terminals are in the peripheral nervous system. Okay, so we're going to talk specifically about the central nervous system, which is filled with those interneurons. And those interneurons are divas. Okay, they need a ton of support and protection. Okay? And then we'll talk about brain structure and function and spinal cord and reflexes. All right, so why do you need so much support and protection for your central nervous system? For one thing, it's super delicate, right? It can be easily damaged, and damaged neurons cannot be regenerated very well, right? They can be regenerated sometimes, depending on what kind of damage it is. If it's just damage at the axon level, potentially grow a new axon, right? But if that cell has completely undergone cell death, or what's called apoptosis, not replacing it. Hey, your brain needs a constant supply of oxygen and glucose. Your neurons only want to do aerobic respiration of glucose. Okay, no anaerobic glycolysis for your neurons. Okay, so they don't do any anaerobic glycolysis. And then they want to run on glucose. They do not want to run on fatty acids, right? And only occasionally can run on ketones. Okay, so if you don't have enough glucose for your brain, it can switch over to running ketones, which your liver has to make from fatty acids for it. Okay, but brain wants glucose. This is why sugar is addictive, okay? Because your brain wants it. Yeah.
So, since neurons can't be regenerated, are babies born with all the neurons they ever have, or do they grow some?
So the general consensus is that you're born with all the neurons you're going to have, although there is possibilities that new neurons are regenerated. As we study the brain more and more, they're finding that there might be some regeneration, right? But it's not a lot. Okay. Yep. It's not a lot. Okay, and then levels of potassium, sodium, and calcium need to be tightly regulated. Why? What do they play a major role in? Neurotransmission, action potentials, right? If you got a bunch of neurons in your central nervous system, right, they're constantly altering their membrane potentials, right? So you have to tightly regulate the key ions that play a major role in resting membrane potential as well as action potentials. Okay, so what we're going to do is we're going to talk about the glial cells, which we briefly mentioned in neurons and synapses. Then we're going to talk about the meninges, then we're going to talk about cerebrospinal fluid, then we're going to talk about the blood-brain barrier, then we're going to talk about this, well, very briefly about the skull and spinal column, right? Basically, we're going to talk about that it's there. All right, so glial cells. 90% of the cells in your central nervous system are support cells. I said neurons are divas. Okay, so every neuron has 9 glial cells. Or for every neuron, there's 9 glial cells. Right, basically the same thing. Okay, so we've got 5 types of glial cells that we're going to talk about. There are astrocytes, which are being shown in green here. And notice that they have these foot processes and they're basically touching everything. Okay. Epididymal cells are here lining the ventricle. Okay, they help to secrete and move around cerebral spinal fluid. Okay, your brain is hollow, right, as is your spinal cord. And the hollow spaces are called the ventricles, and they're filled with cerebral spinal fluid. Okay, microglia are these little purple ones. They're the immune cells of your central nervous system. Because of the blood-brain barrier, your white blood cells can't cross into the brain. Okay, the oligodendrocytes are myelinating the interneurons. So here's an oligodendrocyte. Notice that it myelinates multiple interneurons. And then the glial cells found in your peripheral nervous system are your Schwann cells. And Schwann cells myelinate peripheral neurons, and a Schwann cell will wrap itself completely around a single neuron. Okay, so astrocytes have all those foot processes and basically are sticking their feet in everybody's business, right? They're the main glue of the central nervous system. They hold neurons in place. They play a major role in guiding neurons during fetal brain development, okay, to get the right connections going. Okay, they play a major role in the formation of the blood-brain barrier. They touch the capillaries. And when they touch the capillaries, they tell those capillaries to form tight junctions, to not be leaky. Okay, so the capillaries in your brain aren't leaky like the capillaries in the rest of your body. They play a major role in repairing brain injuries and helping neural scar formation. So if you do damage your brain, they're going to help to repair it. Okay, they play a role in neurotransmitter activity. Remember I mentioned that neurotransmitters can just diffuse away from the synapse, which a lot of them do in the central nervous system. Okay, you don't want that neurotransmitter then going into an adjacent synapse and causing misfiring. So astrocytes take up neurotransmitters once they've diffused away from synapses. Okay, they also take up excess potassium from the extracellular fluid, which is cerebrospinal fluid, okay, to maintain tight control over potassium levels in the cerebrospinal fluid, because potassium is the major intracellular cation. So you don't want to have a buildup of potassium in extracellular fluid. Okay, and along with the other glial cells, they enhance synapse formation and help modify synaptic transmission, which are really important points for learning and memory formation. Okay, so astrocytes have major roles. The other glial cells, we're just going to have a couple bullet points for. So the oligodendrocytes myelinate interneurons. Okay, they myelinate interneurons. You go to add here Schwann cells, myelinate peripheral neurons. Okay, microglia act as the immune cells of the central nervous system. Because our other white blood cells can't cross into the brain because of the blood-brain barrier. Hopefully you aren't fighting an infection in your brain. Okay? If you're not fighting an infection, the microglia still play a role. They release growth factors, okay, to help your neurons and your other glial cells both survive as well as thrive. Okay, so they keep everybody happy. Those epidermal cells line your ventricles. Those are your fluid-filled cavities, are your ventricles. Okay, and they help form and circulate cerebrospinal fluid. Okay. So your CNS is really well protected. Okay, it's enclosed by hard, bony structures, your skull and vertebral column. Then it's also wrapped in 3 protective as well as nourishing membranes, your meninges. You've got your dura mater, the arachnoid mater, and the pia mater. Right? This is closest to the skull, this is closest to the brain. Right? And then your brain is basically floating in cerebrospinal fluid. Okay? And then the blood-brain barrier is limiting what can get into the brain. Okay? So that cerebrospinal fluid can have a completely different makeup than plasma or interstitial fluid. Okay, so here we've got a cross-section, and it's showing the skull. Here's the vertebrae being shown. Okay, and then those 3 meninge layers are being color-coded. The dura mater is being shown as blue, the arachnoid mater is being shown as purple, and the pia mater is being shown as yellow. What I want you to notice is that they're also present, those meninges are present on the spinal cord as well. Okay, the dura mater is closest to the skull. It's super thick and durable, and it has larger blood vessels. Okay, the arachnoid mater is the middle layer, and it's called the arachnoid mater because it has a spider web-like appearance because it's so highly vascularized. So many blood vessels are in the arachnoid mater. Okay, and it's pretty thick, and it provides a nice cushioning effect. Okay, and the subarachnoid space, the space below the arachnoid mater, is filled with cerebral spinal fluid. And the arachnoid villi reabsorb your cerebral spinal fluid. Okay, it's not a stagnant pool. It gets reabsorbed and recycled multiple times a day. Okay, the pia mater is actually attached to the brain as well as spinal cord. Yeah?
Did I hear you correctly that the arachnoid is just all the vascularized nature of the material?
It's highly vascularized. Yeah, so it's got a spider web-like appearance because those are all just blood vessels
Okay, that's what.
that make it look like a spider web.
Okay.
Yep. Uh-huh. Okay, so the pia mater is attached to the brain. It's really delicate. It has the capillaries that form the blood-brain barrier. Okay, and if you get inflammation of the pia mater, that's what meningitis is. Meningitis is. Okay, so meningitis is inflammation of the pia mater in specific. Okay, so here we have a cross-section, and it's showing these are where your arachnoid villi are. We get reabsorption. It's at the top. Okay, and then we have a cross-section showing the vertebrae as well as those 3 meninge layers around the spinal cord as well. Okay, let's talk a little bit about cerebral spinal fluid. It's the extracellular fluid of your central nervous system. Okay, so extracellular fluid can now get separated into 3 compartments that we talked about already. So ECF, extracellular fluid, can be separated into plasma, which is the extracellular fluid inside the cardiovascular system. And then we have interstitial. So interstitial fluid is also known as tissue fluid. That is the extracellular fluid bathing your tissues outside of the central nervous system and cardiovascular system. And then we've got cerebrospinal fluid, which is the extracellular fluid bathing your central nervous system. Okay, and it is secreted by those epidermal cells in the choroid plexus. So here we have a choroid plexus of the 3rd ventricle, right? And we've got a choroid plexus in the 4th ventricle. Okay, so the epidermal cells present in these portions of the 3rd and 4th ventricle are where cerebral spinal fluid is produced. Okay, and then those epidermal cells in the rest of the ventricles help circulate that cerebral spinal fluid, and then it ends up at the arachnoid villi where it gets reabsorbed. Okay, so this is where you get reabsorption, is up here at the arachnoid villi. Okay, and production in the 3rd and 4th ventricles. Plays a major role in cushioning the brain, right? And it also maintains a stable interstitial fluid environment, which is slightly different than plasma. Well, it's quite different from plasma, but it's also different from interstitial fluid outside of the central nervous system. Okay, so here we have the makeup of plasma versus cerebral spinal fluid. Okay, you have a total volume of about 125 to 150 mils. Right? And you produce 400 to 500 mils of CSF a day. So that means you recycle it about 3 times a day. So it's not a stagnant pool of fluid. And it's tightly regulated. Right? So here we've got glucose. So you maintain plasma glucose levels between 80 and 100 milligrams per deciliter so that there's always plenty of glucose in the cerebral spinal fluid. Okay. The fact that glucose levels are lower in the CSF than in plasma, what does that tell you about the transport mechanism for glucose into the brain? Is it active or passive?
Passive.
It's passive, right? Because it's going down a concentration gradient. Okay, so there's a little bit less glucose in CSF. Here, proteins. Plasma is chock full of protein. Not so much in CSF. Okay. Sodium is a little higher in CSF. Potassium is a little lower. The reason why there's a little lower potassium is because those astrocytes take up potassium. Right? Chloride is a little higher. Calcium is a little lower. Right? So this means that CSF has a different makeup than plasma. The reason cerebrospinal fluid can have a different makeup than plasma is because of the blood-brain barrier. Anything that's water soluble normally goes between capillary cells, right? There's normally spaces between capillary cells. And so you're just going to have them move by simple diffusion, and so you'll have a similar amount of glucose in plasma as in interstitial fluid. But because you have tight junctions, that means it has to be transported. Because it's transported, it can be regulated. So that's how you can have different levels of water-soluble substances in CSF compared to plasma. All right, so we are talking about the central nervous system and how well protected it is and how your neurons are divas and have a ton of support cells. And one of the things your central nervous system needs is a constant supply of oxygen. So even though your central nervous system is only 2% of your body weight, it gets 15% of your blood supply. And that is not because it's a conditioning organ. So things like your kidneys get way more blood supply than they need for their metabolic needs. Your brain needs all that blood because it needs the oxygen and the glucose in it, right? Your brain has a really high metabolic rate all the time, Even when you sleep, your brain is super active. You're just not consciously aware of what it's doing, right? So it needs a constant supply of both oxygen and glucose because it doesn't have any glycogen stores, and it only wants to do aerobic respiration. Okay, it does not want to produce any lactic acid. It doesn't use fatty acids like the rest of your body will. Okay, it wants to run off of glucose. And in dire situations, if there's not enough blood glucose, it will use ketones. And your liver makes ketones from fatty acids. Okay, so your liver will make ketones, and then they'll go to your brain, and your brain will run off of ketones. And people talk about when they're on hunger strikes, right? That there's a point where you switch over and you feel euphoric. That might be because of switching over to brain running on ketones. Okay, but you're not going to do your sharpest thinking. Yeah.
Why do diabetics have to get tested?
Oh, okay. So diabetics aren't using glucose properly. So type 1 diabetics that are insulin dependent, they're not producing insulin. So that means their cells won't take up any glucose. Their brain will still take up glucose because your brain is not insulin dependent. Your brain is always taking up glucose. But your other cells won't take up glucose. So they're going to run only on fatty acids. And one of the byproducts of fatty acid oxidation is ketones, right? And ketones are also known as keto acids, so they can alter blood pH, okay? And if you have too many ketones in the blood, you go into ketoacidosis. And you can smell it on someone's breath. Their breath smells like acetone.
So how do you reduce those? Because my brother-in-law got tested and his ketones were too high. How do you?
And he's diabetic?
Yeah.
He needs to control his diabetes better.
Okay.
Yeah.
You said it smells like what?
Nail polish.
Okay.
So someone who's in ketoacidosis, their breath smells like nail polish.
Okay.
Yeah. Okay, so your brain uses 20% of oxygen at rest. When you're exercising, what organ's using a bunch of oxygen? Your muscles, right? Your brain's still using a ton of oxygen, right? It's just that your muscles now are using a bunch as well. And then your brain uses 50% of the glucose at rest. Your brain is super hungry for glucose. That's why sugar is addictive, because your brain loves it, right? And your brain tells you what to do. Right, your brain is protected by what is called the blood-brain barrier. So the capillaries supplying your brain with the oxygen and glucose, instead of being leaky like other capillary beds, have tight junctions. So they aren't leaky. So you have a situation where you have capillaries supplying oxygen and nutrients to your brain, and they're not leaky. Oxygen, how does oxygen get through cells? Just diffuses, right? So it's not like your blood-brain barrier is affecting oxygen levels, because oxygen will just diffuse across those endothelial cells. Water-soluble substances like glucose, amino acids, and the electrolytes would normally go through the pores between the capillary cells. But because there aren't pores, they have to be transported. Because they have to be transported, that means they can be regulated, right? Anything that affects behavior crosses the blood-brain barrier. Okay, so steroid hormones are lipophilic and therefore just diffuse across those endothelial cells. So steroid hormones affect behavior. What are the steroid hormones? The sex steroid hormones. Males have testosterone, and what behavior changes does testosterone cause? So high testosterone leads to aggression, right? And then estrogen and progesterone, the female steroid hormones, cross the blood-brain barrier and affect behavior. Okay, so PMS is due to steroid hormones affecting behavior, right? Pregnancy, you get a huge fluctuation in steroid hormones and behavior changes, right? Anything that affects behavior crosses the blood-brain barrier. Ethanol crosses the blood-brain barrier, okay? Any illicit drug crosses the blood-brain barrier. Right? The blood-brain barrier is formed because of the astrocytes. Okay? The astrocytes tell your capillaries to form tight junctions. So here we have a figure of a regular capillary bed and one of a capillary bed supplying your brain. Okay, so a regular capillary bed, they're only one cell thick, so your capillaries are only one cell thick. Those cells are called endothelial cells. And normally they have pores between them, so they're leaky. Right, so here we have a cross section. So normally you have these pores and small hydrophilic solutes, things like glucose, amino acids, electrolytes, will just go through the pores. Okay, and anything that's hydrophobic will just cross through the endothelial cells. In the blood-brain barrier, those astrocytes, those foot processes, tell those endothelial cells to form tight junctions. Okay, so instead of having pores, you have tight junctions. So that means hydrophilic solutes get transported. Glucose is in higher concentration in the bloodstream than in cerebrospinal fluid. So what type of transport mechanism is used for glucose? If it's in higher concentration in the bloodstream than cerebrospinal fluid.
Passive.
What?
Passive.
Passive, right? And so glucose can't just diffuse through, so it's going to be facilitated diffusion, right? Amino acids, not many amino acids get transported, right? Because your brain likes to use amino acids as neurotransmitters. Okay, but having that blood-brain barrier means that your brain can use really simple molecules as neurotransmitters. Because as those simple molecules fluctuate in the bloodstream, because you just had a meal, etc., it's not going to cause your neurons to misfire if you have that blood-brain barrier. Okay, so we have a blood-brain barrier to prevent chemical fluctuations in the bloodstream from affecting neural transmission. Okay? So fluctuations of chemicals in the bloodstream will not affect neural transmission. It's also going to minimize blood-borne substances that might damage those neurons from reaching them. So remember, they're really delicate and they don't regenerate. Okay, also prevents circulating hormones that also act as neurotransmitters from affecting the brain. Okay, so antidiuretic hormone in the periphery, also known as vasopressin, causes water reabsorption in the kidney. Right? But in the brain, vasopressin acts to help with pair bond formation. Right? So it can have totally different roles in the central nervous system versus in the periphery if you have the blood-brain barrier. Okay? Insulin regulates blood glucose levels in the periphery. Okay? In the central nervous system, it regulates appetite, which is kind of similar, right? But still has a different function in the central nervous system than in the periphery. The problem with the blood-brain barrier, it limits the use of drugs for the treatment of brain and spinal cord disorders. Okay, the drug has to be able to penetrate the blood-brain barrier in order to have an effect. So it has to be lipophilic. Otherwise, you have to inject it directly in, and no one wants that. Right? Okay. Any questions about supporting and protecting your central nervous system? Right, then let's start talking about brain structure and function. Hey, so why are we even talking about the brain? What has it done for you lately, right? Is it just a vestigial organ that you can get rid of, right? Your brain obviously plays a huge role in physiology. So it regulates your internal environment, plays a huge role in homeostasis. And we'll talk about the hypothalamus, okay? And we'll talk about it when we talk about the nervous system, because it is part of your brain. And then we're going to also talk about the hypothalamus when we talk about the endocrine system a little later in the semester, because your hypothalamus is the direct connection between your nervous system and your endocrine system, and it regulates homeostasis. Okay? Your brain allows you to experience emotions. Hopefully that emotion is joy right now from learning physiology. Okay? It allows you to control your movements, right? We have conscious control over our skeletal muscle. Allows us to be consciously aware of our own body and our surroundings, right? And then it allows us to do higher cognitive processes like thought and memory formation, okay? Learning, math, language, science, right? That's all higher functions of your brain. Okay, so we're going to go through some of the basic structures in their brain and their major functions. Okay, if this is really interesting stuff to you, I recommend looking into some of the neuroscience courses where you'll go into more depth on various areas of the brain. Right, but for now, we're just going to cover the major brain areas and their major functions. Okay, so we're going to talk about the brainstem, the cerebellum, and then we're going to move up to the forebrain and separate it into the diencephalon and the cerebrum. The diencephalon can be separated into the hypothalamus and the thalamus, and the cerebrum can be separated into the basal nuclei and the cerebral cortex. Okay, and they're listed in order from oldest evolutionarily to newest. Okay, so what makes us human is the fact that our cerebral cortex is large and really complex. Okay, so our humanness comes from having evolved a really large, complex cerebral cortex, not because we've got an awesome brainstem, right? Right, so here we have a brain with a mid-sagittal view, and we've got the forebrain. Here's the cerebrum, okay, and then the diencephalon can be separated into the thalamus and the hypothalamus. And notice the hypothalamus is directly connected to the pituitary gland, which is part of the endocrine system. So I said it's the direct connection between the nervous system and the pituitary glands. Okay, and then being shown here is the corpus callosum, because we have two hemispheres of our brain, right and left. They got to talk to each other, and they talk to each other via the corpus callosum. Okay. And then we have our cerebellum at the base, and then we have our brainstem, which is the midbrain, the pons, the medulla oblongata. The brainstem is directly connected to the spinal cord. Okay, so the brainstem is the oldest region of the brain. It was the first portion of the central nervous system to evolve, and it's continuous with the spinal cord. Okay, and it links the rest of the brain to the spinal cord. Okay, we're going to talk about the midbrain, the pons, and the medulla in the brainstem. Okay, so the brainstem, most of your cranial nerves arise from the brainstem. You don't have to memorize them for this class. Okay. In the brainstem, we also have the reticular formation, which is being shown as this blue line here. Okay, notice that the cerebrum is missing from this brain. Okay. The reticular formation receives and integrates incoming sensory information, meaning there's synapses there between afferent neurons and interneurons. And for most sensory information, this is the first synapse. Okay? For sensory information, except for smell and vision, all your other sensory information is going to synapse first in the reticular formation, so in the brainstem. Okay. The areas that govern sleep are also in the reticular formation. Okay, and the areas that govern sleep have inhibitory interneurons. And basically, they're inhibiting sensory information from leaving the brainstem because you have no consciousness in your brainstem. Okay, you are not consciously aware of any information synapsing or going on in your brainstem. Okay, it's subconscious. Okay, so if you inhibit sensory information at the level of the brainstem, you never become consciously aware of it, and you can sleep. Okay. Vision doesn't synapse in the reticular formation, but what do you do to stop visual information when you sleep? You close your eyes, exactly. Okay, your brainstem controls heart and blood vessel function. Okay, things that you subconsciously control. Right? Also controls respiration, although your diaphragm is a skeletal muscle, right? So you can consciously override your brainstem for respiration. So one of the ways to calm yourself is to take slow, deep breaths, because you can't slow your heart rate consciously, but you can slow your respiration rate consciously, and that will help to slow your heart rate. Okay, and then lots of digestive functions get controlled by your brainstem. Okay, so this is all subconscious. Brainstem also helps to regulate muscle reflexes involved in equilibrium and posture, so that you don't have to waste conscious effort on sitting up straight. Although some of you should waste some conscious effort on it. Got lots of schlumpers in here. Okay. Right, so most of your cranial nerve, oh yeah, question.
I just want to make sure I heard you correctly. The reticular formation is where you have the first cycle of sensory input. Was that correct?
Reticular formation is where you have the first synapses between afferent neurons and interneurons. Okay, so the reticular formation, afferent neurons for your somatic senses, okay, for taste, for hearing, synapse first in the reticular formation with interneurons. Okay.
Thank you.
Yep. Okay, one of the roles of your brainstem is that it's the origination for most of your cranial nerves. I'm not showing you this so that you memorize all your cranial nerves, but just as a reminder to tell you guys that nerves are bundles of axons. Okay? Nerves are bundles of axons, and they contain both afferent and efferent neurons in there. Okay? So nerves are bundles of axons that contain both afferents and efferent. So they supply both sensory afferent neurons running towards the brain, as well as motor neurons running away from the brain. Okay? And then the reticular formation is the first stop for most of your sensory information, except for vision and smell. Okay, let's move up a little bit. Talk about the cerebellum. So the cerebellum is attached at the rear portion of your brainstem. It's being shown in this darker pink color. Your cerebellum contains more individual neurons than the rest of your brain. Right? There are a ton of neurons in your cerebellum. Okay? And the role of your cerebellum is to maintain proper position of your body in space. Okay, so it plays a major role in equilibrium, postural muscle contraction. Okay, it regulates subconscious motor activity. Right? So this is motor activity you don't have to consciously coordinate anymore. These are learned motor reflexes for things like walking. Right? All you have to think about is watching a toddler learning to walk. Right? They have to expend an enormous amount of mental effort to walk because it hasn't become a learned reflex yet. And once it becomes a learned reflex, you're in trouble, right? Okay? For you guys, going out and walking up those stairs, as long as you weren't texting, usually you can completely master it, right? Without much conscious effort. And that's because your cerebellum is controlling those motor reflexes, those learned motor skills. If you memorize a piece of music, right, where you just sit down and you play it, that's held in your cerebellum. Okay? If you are an athlete, say you're a tennis player, you know, your tennis serve is held in your cerebellum, right? Let's move up now to the forebrain. And we're going to talk about the Diencephalon first. Okay, so the Diencephalon houses your thalamus and your hypothalamus. Okay, so here's the cerebellum. Here's the brainstem. So we've got the inverse image of the brain. Here is the cerebrum, and we're seeing the thalamus and the hypothalamus. Okay, the thalamus performs some primitive sensory processing. So we have our first synaptic input between afferent neurons and interneurons in the reticular formation. Interneurons then run from the reticular formation, or the brainstem, up to the thalamus, and we have a second round of synapses. Okay? And you get some crude awareness at the thalamus. So someone who's in a coma or is said to be brain dead may occasionally respond to sensory information because their thalamus is still functioning, but because their cortex is not functioning, they're not truly consciously aware of it. Right? Your hypothalamus controls homeostasis. And it's the only part of your central nervous system that doesn't have the blood-brain barrier present. Because it needs to know what's going on in the rest of the body. Okay? It needs to know the osmolarity of your bloodstream. It needs to know the pH, etc. Okay? So it doesn't want the blood-brain barrier. It wants to know what's happening in the bloodstream. Okay, so the thalamus has a series of synapses for sensory input. Okay, all sensory information except for olfaction has synapses in the thalamus. So this means vision synapses in the thalamus. Remember, vision does not synapse in the brainstem, but it does synapse in the thalamus. Okay, and the thalamus serves as what's called a relay station. It determines, it's the last stop before sensory information hits your cortex. Once it hits your cortex, you become consciously aware of it. So your thalamus plays a really important role in only sending up the most important information. Okay, so that you only pay attention to important stimuli, right? So that you can focus. So you can do crude awareness at the thalamus level. However, location and intensity are determined in the cortex. Okay, so it would just be really crude awareness. Okay, you haven't hit true consciousness yet. Okay, the hypothalamus plays a major role in regulating the internal environment. And look at all the things it regulates. It regulates your body temperature. It regulates thirst and urine output via the hormone, antidiuretic hormone. Okay, it regulates food intake. Okay, it controls anterior pituitary hormone secretion. And it produces the hormones that the posterior pituitary releases. Okay, so it is the direct link between the nervous system and the endocrine system. It controls uterine contractions and milk ejection via the hormone oxytocin that the posterior pituitary releases. It serves as the major autonomic nervous system coordinating center. So this is fight or flight, rest and digest. Sympathetic and parasympathetic. Your hypothalamus controls your sympathetic and parasympathetic nervous system, which we'll talk about at the end of the week. Okay, your hypothalamus is part of what's called your limbic system, which is your emotional center. So it plays a major role in emotional and behavioral patterns. Okay, and it also plays a major role in your sleep-wake cycle. So your hypothalamus is pretty important. It helps regulate homeostasis. And again, we'll talk about when we talk about the endocrine system. All right, so let's move up to the newest region of the brain, the portion of the brain that makes us human. And we'll spend a little more time talking about the cerebrum. So in humans, our cerebrum is highly developed. Okay, and it makes up 80% of our total brain weight. Although remember, our cerebrum has way more neurons. Okay, our cerebellum. Our cerebellum has way more neurons than the cerebrum. Our cerebrum has way more connections between neurons. So a big portion of our cerebrum's weight is myelinated axons. Okay, our cerebrum is where we get gray versus white matter. So the gray matter is cell bodies of inner neurons. Our white matter is the myelinated axons. Okay, we'll look at that distribution of gray versus white matter. Yeah?
Just a speculative question.
Yeah.
If you have someone that has a profession that is very physically oriented, say a ballet dancer,
Uh-huh. Yeah.
that ratio, would you see that change as far as the number of connections to where they start to even out a little bit?
The number of, you mean in the cerebellum versus the cerebrum?
Right.
No, because you don't make new neurons.
Oh,
Yep.
I was thinking.
Yep, so early in development, you just have way more neurons in the cerebellum, and you retain that number of neurons throughout development. You can see new connections potentially being made in the cerebrum, right? But you're not going to be making new neurons in the cerebrum,
Oh,
if that makes sense. Yeah. Okay, so we also have the basal nuclei, which is part of the cerebrum, and we'll look at where those are housed. Okay, but the outer surface, the cerebral cortex, is this highly convoluted area of gray matter. Okay, so the outer portion of the cerebrum is your cerebral cortex. It's made up of gray matter, and it's the most complex integrating area of the brain. And it's where your really sophisticated neural functions come from. So thought, memory, language, etc. are all coming from the cerebral cortex. Yep.
You said the white matter is the myelination of the axons?
Mm-hmm.
Okay.
Yep. White matter is myelinated axons. We'll look at that. We got to figure. Okay, so gray versus white matter. This is what's showing gray versus white matter. Here we've got gray matter. It's just synapses and cell bodies. Okay, so gray matter is synapses and cell bodies of interneurons. Everything's an interneuron up in the cerebral cortex. And notice, so the gray, I don't know why they color-coded the gray matter tan. Okay, unless I don't know who would say this is gray at all. Okay, but the gray matter in this dark tan color is on the outer portion. Okay, that's the cerebral cortex. Okay, the white matter is the myelinated axons. Myelin is mainly lipid. What color is lard or shortening? White. Okay, so that's why white matter is white. It's primarily lipid. It's all those myelinated axons. So what this is showing is we've got the gray matter on the outer areas, and then we've got white matter, and then all these purple lines are showing all these tracks of axons running between different parts of the brain. So we have association fibers. Oh, here in blue, we'll wait for the jet to pass, right? Here in blue is all those myelinated interneurons in the corpus callosum. Okay, so those are running between the hemispheres. That's why they look like they got cut off. Okay, so the little blue dots are all those myelinated interneurons running between the corpus callosum, connecting the two hemispheres, right? And then the projection fibers are being shown in these dark blue, and those are running up and down, okay, from the cerebral cortex to other parts of the brain. Okay, so the association fibers are running between areas of the cerebral cortex within one hemisphere. Okay, commissural fibers are part of the corpus callosum running between the hemispheres. And then we've got the projection fibers that are running between the different brain parts. Okay, and they're all just being color-coded a different color. In this figure, what we see is we have areas of gray matter within the white matter. So here we've got the thalamus. Okay, it's areas of gray matter within the white matter. Okay, and then we have the basal nuclei that we're going to talk about. So the basal nuclei are areas of gray matter within the white matter of the cerebrum, or a pocket of gray matter, you can think of it. Okay. So lots and lots of connections is the take-home message. Loads of myelin. Okay, so the basal nuclei are those pockets. So again, here we've got that cross-section, the gray matter on the outside, the white matter. Here we've got the thalamus. Here we have some pockets of gray matter for the hypothalamus. And here are the basal nuclei. You don't need to memorize the names of them. Okay, so the basal nuclei are pockets of gray matter within the cerebrum. And they modify motor pathways, descending motor pathways. Okay? The basal nuclei modify descending motor pathways. And a great way to visualize what the heck basal nuclei do is to know that they are damaged in Parkinson's disease. So Parkinson's disease is a disorder of your basal nuclei. Your basal nuclei have inhibitory interneurons in there. And they're trying to inhibit descending motor pathways so you get smooth movements. No herky-jerkies, okay? No tremors. People with Parkinson's disease, not only do they have tremors, but oftentimes their muscles are super stiff. So it seems like they're really slow, but it's actually that their muscles are already contracted. So it's hard for them to stand up because their muscles are already contracted. Okay, so people with Parkinson's have tremors as well as overly stiff muscles. And that's because their basal nuclei are not functioning properly. They're not inhibiting that descending motor information. Okay, so let's talk about that cerebral cortex a little bit. So this is the newest region of the brain evolutionarily. It's what makes us human. Right? It's where we get the most complex integrating areas of our brain and where we get our sophisticated neural functions, like voluntary initiation of movement, which maybe doesn't seem like a big deal. Lots of other animals initiate movement, right? Okay, sensory perception. So we get our final sensory perception for auditory information, visual information, taste, smell, somatosensory. Okay, it's where you have your conscious thoughts coming from. Right? It's where our language centers are. That's something that makes us very human, is we have a very rich language center. Okay, it's where our personality traits reside. Okay, and it's where your intellect resides. Okay, so we already talked about how we have two hemispheres. So we have a right and left hemisphere. Especially important, we have a right cerebral cortex and a left cerebral cortex. These hemispheres are connected by the corpus callosum. Okay, so they don't work independently of one another. The corpus callosum is 300 million axons running between your two hemispheres. So loads and loads of crosstalk. Okay? Each hemisphere can then be separated into 4 lobes, and each has a specialized function. Okay, so we have the occipital lobe, which is at the back, and it's for visual information. We've got the temporal lobe under the temples, and that's for hearing information. And then we've got the parietal lobe, which is sort of at the top of the head, right? And that's where we get somatosensory information. And then the frontal lobe is at the front, right? And that's where you get voluntary movement coming from, as well as personality traits and intellect from the frontal lobe. Okay, so here we've got a brain. We've separated into the right hemisphere versus left hemisphere, and then we're showing those 4 lobes. So purple is the frontal lobe, parietal is the blue lobe, occipital is the green lobe, and yellow is the temporal lobe. And what's this structure right here? The cerebellum. Okay, so the hemispheres do different things. So the left hemisphere is specialized for logical, analytical, sequential, and verbal tasks. So this is where your language centers are. They're in your left hemisphere. Okay, it's also where you math from. Okay, people who are super into philosophy also are considered left-brained. So a lot of you in this room are probably considered left-brained if you're into science, right? Okay, the right cerebral hemisphere excels in non-language skills, so things like spatial perception, artistic, musical talents. Okay, so creative people are said to be more right-brained, right? Some of you are probably really balanced, etc. Okay, but you do have some specialization within the 2 hemispheres. But remember, there's a load of crosstalk because of the corpus callosum. Okay. So let's just discuss each of those lobes and talk briefly about their major functions. So the occipital lobe, being shown in green, carries out visual processing. So you can think you have eyes in the back of your head, right? The temporal lobe, shown in yellow, does the initial reception of sound sensation. Okay, it's right under your ears. The parietal lobe does somatosensory processing, right? So that's information coming from your skin surface. And then the frontal lobe does voluntary motor activity, speaking ability, and elaboration of thought, right? So higher level thought processes occur in the frontal lobe. It's also the personality center. Okay, so when people used to have aberrant personality traits, they would do frontal lobectomies, where they basically just destroy parts of the frontal lobe, and usually then that person would just be, you know, kind of zombie-like. Not done anymore, at least not by, you know, ethical physicians. Okay, so let's talk about specialized areas within each of these lobes. We're going to talk about the somatosensory cortex first, because it's pretty well understood. It's the front portion of each parietal lobe, so it lies really close to the frontal lobe. Okay, and it does initial processing for your somesthetic sensations. So that's anything coming from the skin surface. So pressure, heat, cold, as well as pain. Right? It can localize the source of sensory input and determine the intensity. So it can determine where and how bad. Right? It receives input from the opposite side of the body. So your somatosensory cortex in your left parietal lobe does information coming from the right side of your body, and the somatosensory cortex in your right parietal lobe does information coming from the left side of your body, because those projection fibers cross in the spinal cord. It is also called the sensory homunculus, which means little man. And so let's actually look at the sensory homunculus. Although first we're going to do the motor cortex. I always get tripped up. I always expect it to be the figure next. Let's talk about the motor cortex, because then we're going to look at a figure of the motor cortex and the somatosensory cortex together. Okay, so the motor cortex is in the frontal lobe, and it does voluntary control over your skeletal muscles. And it also has a map. Right? And it also controls the opposite side of the body, because again, those projection fibers cross. And it can be depicted by the motor homunculus. And so now let's look at the somatosensory and the motor homunculus. Okay, so here we've got someone with their skull removed, right? Question?
Yes. Is there like an evolutionary benefit to having it cross?
That's just what happens during development, is that there's crossing of those fibers in the spinal cord. That's just how it evolved. I don't know if there's any particular benefit. That's just what the system is. Yep. Okay. So here we've got the parietal lobe versus the frontal lobe. So the green is showing you where the somatosensory cortex is in the parietal lobe, and the blue is showing you where the motor cortex is in the frontal lobe. So you notice they lie close to one another. Okay, and here's what your somatosensory cortex thinks you look like. Right? You're all face and lips and hands. The amount of area your somatosensory cortex devotes to a specific area of your body tells you something about how many afferent neurons are arising from that area of the body. Okay, so we are very tactile. We use our hands. Okay, and then our lips are super sensitive as well. Okay, so our fingertips and our lips have really high acuity, meaning we can detect 2 points that are really close together, because we have a ton of sensory neurons coming from those areas of the body. Now if we look at the motor homunculus, or the motor cortex, Okay, we also have a huge amount of area devoted to our face and our lips as well as our hands. And the amount of area our motor cortex devotes to a specific area of body tells you about fine motor skills. Okay, so wherever you have a large area for the motor cortex, you have a lot of motor neurons going to that area of the body. And when you have a lot of motor neurons going to an area of your body, that means you have fine motor control. Okay, so moving our hands and our lips and our tongue takes fine motor control, right? To move our lower limbs, that's gross motor control. You only need a few motor neurons to move those really big skeletal muscles. Okay, you need loads of motor neurons to move the fine, tiny ones. All right, we'll stop there for today. Right, so we are talking about different specialized areas of the cortex, and one of those specialized areas is for language, right? So we have a Broca's area and a Wernicke's area that are our language centers. Okay, the Broca's area governs our speaking ability, right? To be able to do the mouth movements and tongue movements that allow us to have coherent speech, whereas the Wernicke's area is for language comprehension, right? And it tells the Broca's area what to say. Okay, so you notice we've got these black lines running between the Wernicke's area and the Broca's area, and that's because the Wernicke's area is telling the Broca's area what to say to make a coherent pattern of speech. These areas sometimes get damaged, right? Or there's issues with these areas that lead to language disorders. So aphasias. What's an aphasia? Inability to speak. Okay, so you guys might have these momentarily. You know, there's a word you know, but you can't think of it, right? And as you age, they get worse and worse, okay? Strokes can cause much more serious aphasias, right? There are speech impediments, so lisps, stutters, okay? Those would be speech impediments. And then dyslexia. What's dyslexia? Someone who's dyslexic does what? Has a problem reading, right? They sort of transpose letters, etc., words. Okay, so which one do you think is an issue with the Broca's area? What? No. Speech impediments. Okay, aphasia is your Wernicke's area having a problem coming up with the word for your Broca's area to say. Okay? A speech impediment like a lisp or a stutter is an issue with the Broca's area. Okay? How about dyslexia? That's going to be an issue with the Wernicke's area because it's all about language comprehension. Okay? Right, so here we've got the brain, and it's being all divvied up into specific functional areas. Okay, so we talked about the primary somatosensory cortex and the primary motor cortex. Okay, so you have to know about those two association areas. Okay, and then the premotor cortex just helps coordinate what the primary motor cortex wants to do. Right, the prefrontal association areas are where you have ideas. Okay, it's your intellect resides there, personality in that prefrontal association area. Just talked about the Broca's area for forming words. Notice that it's close to the mouth of the primary motor cortex. Okay, so that's why it's helping to control that fine motor control of your mouth. Okay, the olfactory cortex we'll talk about when we talk about the sensory systems. Okay, the olfactory cortex is for your sense of smell. We'll talk in a little bit about the limbic association cortex. It actually is a bunch of different brain structures. We have a better figure to show that. The primary auditory cortex is for sound perception. We'll talk about that when we talk about sensory systems. Auditory association areas are within that auditory cortex, and notice that it lies really close to the Wernicke's area. Okay, so when you're hearing words, your Wernicke's area gets involved in order to comprehend what's being said. Okay, and then we've got primary visual cortex for sight perception, and then association areas for vision, in order to decide what the heck you are actually seeing. Okay, and then some sensory association areas, mainly for the somatosensory information coming in. Okay, so there's a bunch of different association areas that have specialized function. Yes?
For the exam, do we need to point these out where they are?
No, you'll just need to know the major roles of the primary somatosensory cortex, the primary motor cortex. So the ones that have separate slides. Okay? This is just showing that there's not blank areas in your brain. Okay? There's lots of different specialized areas of the cortex. But you need to know the function of the association areas that have separate slides in the lecture. Like the limbic association cortex. So this figure is a little better, so it's showing all the brain structures involved in the limbic association cortex. It's highlighting them in green. Okay, notice the olfactory bulbs are involved, the hypothalamus is involved, the thalamus is involved, the amygdala and hippocampus. Okay, the amygdala is your fear center. Okay, the hippocampus is where you store your memories. All these areas are involved in the limbic association area. And this is your emotional center of your brain. Okay, and this is where basic inborn behavioral patterns related to the survival of the species come from, i.e. sex drive, right? Also plays a huge role in motivation and learning, okay, and memory formation. Okay, fearful experiences make much stronger memories. Okay, smell. Notice that the olfactory bulbs are associated with the limbic association cortex. Okay, when you smell vanilla, that might, evoke all these fond memories of having cookies being baked at home, right? So smell often triggers pretty strong memories because the olfaction system is part of the limbic system. Okay, how do we know anything about the brain? Okay, a lot of what we know about the brain is either from EEGs or PET scans, right? Or CT scans or MRIs. Okay, especially about the human brain. We're not sticking probes in and seeing what happens. Okay. A lot of studies of other vertebrate species, lab animals, etc. They can do that, but it's not ethical to do that to humans, right? And so a lot of what you know about the human brain comes from electroencephalograms, which you guys did in lab, right? Okay, you only did a 3-lead one, so it wasn't super high fidelity. Okay, but the EEG was actually recording action potential, so electrical activity that was occurring in the brain. Okay, and you get brain waves as an output for EEGs. And we'll look at some example EEGs. Okay, PET scans are often also used. And what PET scans are is they are scans that detect the magnitude of blood flow. So usually you get injected with radioactive glucose, right? And because the brain only runs on glucose, that radioactive glucose is going to go to the brain and it's going to increase in areas that are getting higher blood flow, and those areas that are getting higher blood flow are active at that time because neurons that are firing action potentials have a greater need for glucose. Okay, so PET scans show those images where you get areas of the brain lighting up, and we'll look at some PET scans as well. Okay, so EEGs give output that are called brain waves. So here we've got EEGs from someone who's awake and alert, where beta waves are dominating. Here we have someone who's awake with their eyes closed. So that's what resting means, eyes closed. And it's dominated by alpha waves. Okay, which ones were higher frequency, alpha or beta? Who remembers? Beta are higher frequency. Okay, so even though it looks like less is going on, because the waves look smaller, it's actually because there's such high frequency waves that they're desynchronizing. Okay, so when you are eyes open, awake, loads of stuff is happening in your brain. Okay, and those waves, those beta waves, are super high frequency, so they end up canceling each other out. So it doesn't look like a whole lot is happening. Okay, when you close your eyes, you're not getting visual information anymore, and we're a super visual species. So then the lower frequency alpha waves dominate. Hi, and then we have stage 2 sleep, stage 3 sleep, stage 4 sleep, and REM sleep. Okay, so as you go through the different stages of sleep, the different brain waves take over. So you start getting these K complexes, that delta waves dominate, so you get a lot of synchronization within the brain during the deep stages of sleep. And then you have REM, or rapid eye movement sleep, interspersed between those deep stages of sleep. And what does REM sleep look like? Which does it look like? Beta, alpha, beta. Okay, so when you're in REM sleep, or rapid eye movement sleep, your brain looks like it's awake. Okay? That's when you're dreaming. And if you come upon someone sleeping and their eyes are moving under their eyelids, they're in REM sleep. Okay? And that's what's called the paradoxical sleep, because you're sleeping, but you look like you're awake. Okay? So if you have a sleep study done, they're looking to see that you go through all the appropriate stages of sleep and that you get enough of each type. Okay, PET scans are really nice to figure out what areas of the brain are involved in different functions. Okay, so this is from a human biology textbook where they superimposed a PET scan onto this guy's head, right? This is not what your PET scan would look like. It wouldn't be your face in profile with your brain being shown up. Okay, so here's a guy who is having a PET scan done, and he is simply hearing words. What areas of his brain are lighting up? So his eyes are closed, right? So what areas? The darker color is temporal lobe. And what's in the temporal lobe? Auditory cortex. Okay? What else is there for language comprehension? The Wernicke's area is lighting up too. So he's understanding those words. Okay, now they told him to say something. What area of the brain's lighting up? Broca's area. Okay, seeing words. What area is lighting up? Occipital lobe. So he is probably looking at words he can't understand. And how do I know that? Because what area is not lighting up when he's seeing words? Wernicke's, right? So he's not understanding the words he's looking at. Okay, whereas look at what happens when he's actually reading words. Okay, then we get some of the Wernicke's area lighting up and the somatosensory cortex as well. Okay, so reading involves a lot of coordination. Okay, any questions about brain structure and function before we talk about the spinal cord? Okay. So your spinal cord, like your brain, is hollow. So it's a cylinder of nerve tissue. It's continuous with your brain stem, so it's continuous with your brain. And it's highly protected by the vertebral column. And it's the origin of all your spinal nerves. So there's 31 pairs of spinal nerves. We've got our cervical nerves, we've got our thoracic nerves, we've got our lumbar nerves, and we've got our sacral nerves. Okay? And the cervical, thoracic, lumbar, and sacral nerves will come into play when we talk about parasympathetic and sympathetic nervous system. Okay, because the sympathetic nervous system arises from our thoracic and lumbar regions, and the parasympathetic from the cervical and sacral. Right, so what this figure is showing is where all those nerves are running. And again, just a reminder that nerves are bundles of neurons, and they contain both afferent and efferent neurons. Okay, so your, let's say, thoracic nerve 1 is having afferent somatosensory neurons running from this area of the body, right, as well as controlling skeletal muscles in that area. Okay, so afferent and efferent. Okay, so here we have a cross section of the spinal cord, and we've separated it into the dorsal and the ventral half. Okay. And the way I like to remember this is days. So the dorsal half of the spinal cord is carrying afferent info. Okay, so it's sending tracks to the brain. Right? And then the ventral half is carrying efferent information. So information coming from the brain going to the body. Okay. So same is sensory is afferent information. That's going to the brain. And motor is efferent information. And that is going down the spinal cord to the rest of the body. Okay, the spinal cord is also made up of gray and white matter. And so this dark tan color is the gray matter, right? And this lighter tan color is the white matter. I don't know why they couldn't make it gray and white, okay? So the gray matter is cell bodies and synapses. The white matter is myelinated axons. So in the dorsal half, those are myelinated axons running towards the brain. In the ventral half, those are myelinated axons running from the brain. Okay, here we've got a spinal nerve, which again is bundles of afferent and efferent neurons. And the sensory neurons, we have a sensory receptor in the skin, so it has specialized endings of its dendrites. And then we've got the axon hillock, and then the axon, and we get the cell body occurring not with the dendrites. Okay, the cell body is occurring what's called the dorsal root ganglion. A ganglion is any time you have a cell body of a neuron outside of the nervous system. Okay, and we're going to talk about ganglions with the parasympathetic and sympathetic nervous system as well. So they have cell bodies that occur outside of the central nervous system. Okay, so afferent neurons, their cell bodies occur in the dorsal root ganglion. And then they're going to synapse on interneurons within the gray matter of the dorsal half of the spinal cord. And then the efferent neurons, their cell bodies, the motor neurons, are held within the gray matter of the ventral half. Okay, and then the axon runs out in synapses on the effector organ. If it's a motor neuron, that is going to be skeletal muscle. In a little bit, we'll talk about parasympathetic and sympathetic nervous system, which can control glands and smooth muscle as well as cardiac muscle. Okay, so the white matter has tracks, ascending versus descending. Okay, so based on what I told you, which half, dorsal or ventral, has ascending tracks? Dorsal. Okay, so that means ventral has descending tracks. Okay, ascending is afferent, descending is efferent. Okay, the physiology of the spinal cord we're going to talk about is reflexes. Although not all reflexes are held in your spinal cord. Okay, we're just going to talk about a few simple ones that are. So a reflex is any response that occurs autonomically. You don't have conscious effort. Okay, it just happens without your consciousness being involved. And there's two main types. We're going to talk about the physiology of simple or basic reflexes, but there's also acquired or conditioned reflexes. So simple or basic reflexes are unlearned. Okay, these are stretch reflexes. That's what happens when you hit the patellar tendon with a reflex hammer. Okay, you don't have to learn how to do that. Your leg's just going to kick out because of a stretch reflex. Okay, withdrawal and crossed extensor reflexes, that's what happens when you step on a nail, right? You automatically withdraw your foot before you even realize it. Okay, and then a withdrawal reflex would be if you, or a simple withdrawal reflex would be if you touch something hot. You just automatically withdraw your hand. Okay, you don't have to learn that. Okay, acquired or conditioned reflexes are the result of practice and learning. So walking is an acquired reflex. You had to practice to do it. Okay, so complex motor pathways that you don't consciously think about anymore are acquired reflexes. Okay, and where are they held? What brain area? Not the spinal cord. What's the brain area that holds our learned or acquired reflexes? The cerebellum, exactly. Okay, so we're going to talk about a reflex arc and then talk about a stretch reflex and a crossed extension withdrawal reflex. Okay, and both have simple reflex arcs. So the 5 basic components of the reflex arc is you have to have some sort of stimulus. Okay, if we're talking about the knee-jerk reflex, what's the stimulus? Did you say pain? I hope it's not pain. You had a crappy lab partner if it was pain. The stretching of the patellar tendon by the reflex hammer. Okay, and then there was a sensory receptor, which is a stretch receptor that is actually in your quadriceps. Okay, you're stretching your tendon, which then stretches your quadriceps. And your skeletal muscles have these stretch receptors to prevent them from being pulled. Okay, and so when the stretch receptor fires an action potential, it goes up the afferent neuron and it synapses in the spinal cord. So in this case, the integrating center is the spinal cord. Okay, and then that spinal cord is going to send a signal via the efferent neuron, which will be a motor neuron. Okay, and it'll synapse onto skeletal muscle. That skeletal muscle will be in your quadricep, and it will tell your quadricep to contract, which leads to the response of your leg kicking out. Okay. So here we have a diagram showing that simple stretch reflex. So with the reflex hammer, you're supposed to hit the patellar tendon, right? And not super hard. Okay. Okay, so you hit the patellar tendon, that stretches the quadriceps, which has stretch receptors in it. Okay, and for simplification, we're showing 1 afferent neuron and only 2 efferent neurons. More than one are involved, right? But we're just showing for simplification. A single afferent neuron that's attached to the stretch receptor fires an action potential. Okay, here's the cell body in the dorsal root ganglion. It's going to enter and synapse in the gray matter of the dorsal half of the spinal cord. Okay, it's going to send up a synaptic processes towards the brain, and it's going to synapse in the reticular formation. Okay, and then it's going to send another neuron to the thalamus, and then it's going to send a third neuron up to the somatosensory cortex. So you will become consciously aware of what the heck's happening on your leg. Okay? However, before you're even consciously aware, before you get a signal up in the somatosensory cortex, okay, the afferent neuron has synapsed directly onto an efferent neuron. So this is a really simple reflex arc. And that efferent neuron is a motor neuron, and it sends a signal to your quadriceps to contract. Okay, it also synapses on an inhibitory interneuron. This interneuron is color-coded red because it's inhibitory, and it inhibits the efferent neuron or the motor neuron that's controlling your hamstring so that your hamstring does not contract. Okay, so your quadricep contracts, your hamstring relaxes, your leg kicks out. Okay, and the reason for this reflex is to prevent the sarcomeres, or the actin and myosin, that's the contractile unit in your quadricep, from being stretched apart. Okay. All right, any question about the simple stretch reflex? Okay, so skeletal muscles have stretch reflexes. In response to being stretched, they contract. And they're very simple reflexes. Okay, a little more complicated reflex is the withdrawal and crossed extensor reflex. And this happens on your lower limbs only. Okay, if you touch something hot, you just have a withdrawal reflex. Okay, on your lower limbs, if you step on something, say a nail, that sets off action potentials in nociceptors. Nociceptors are your pain receptors. Okay, so the nociceptor acts as the detector of the stimulus. Okay, it fires an action potential, runs up the afferent neuron, enters the dorsal half of the spinal cord, and then it has a series of synapses. Okay, it synapses on a couple inhibitory interneurons, and then it synapses on a couple excitatory interneurons, and so it's going to block signals to some muscles, and it's going to cause other ones to contract. And so what it's doing is it's going to cause the leg that has stepped on the damaging object to withdraw, right? And at the same time, it's going to cause your opposite leg to get ready to hold all your weight. Right? Because the last thing you want to do is withdraw that foot and then just fall over, right? Okay. A withdrawal reflex in your upper limbs is a lot more simple, okay? It's just going to pull your hand away. You're not then going to punch with your other, right? Or maybe you will, but that's probably more of a learned reflex. Okay. Any questions about reflexes? Yeah.
So withdrawal reflexes happen in upper and lower limbs?
Yes, withdrawal reflexes happen in upper and lower. It's just that only the crossed extensor withdrawal reflex happens in your lower limbs. Okay. Yeah.
So does that cross extensor reflex happen when you step on a nail?
It happens when you step on a nail, or if you step on anything and need to withdraw your foot quickly. Glass, fire. Did someone say fire? A Lego, exactly. I've stepped on many a Lego. Yeah.
Okay, so we just explain that cross extensor.
So the crossed extensor, so it's crossed extensor means that it's only in your lower limbs. So it's going to tell the limb that stepped on the object to withdraw, and then it's going to tell the opposite limb to get ready to hold all your weight. So it's involving both limbs. Whereas the simple withdrawal reflex that you get in your upper limbs, when you touch something hot, that hand pulls away. The other arm doesn't do anything. Yeah. Okay, any other questions? All right, let's start talking about peripheral nervous system.